Testing. We're on. We're live. Okay, we're about to get started in our next session, and I'm going to turn it over to Tobias Hemp, who's going to introduce our next keynote. Tobias.
Yeah, thank you for the introduction, and thank you for the invitation to host this next session called The Applications of AI and Machine Learning for Genomics and Global Health. I myself am a senior manager at Illumina in Carl Farr's AI lab. We have developed many AI tools to predict the impact of variants over the years, but there's the same problem as everybody else, namely how to bring them into the hands of the people who could benefit from them the most. I could therefore not be more excited about this next session with a lineup of speakers who might actually have the answer to this and similar questions. Without further ado, I'd like to introduce our first keynote speaker, Ewan Birney. He is the interim executive director at the EMBL. Previously was the director of the EBI and deputy director general of the EMBL. In the past, he has led projects such as the analysis of the human genome gene set and the ENCODE project. Please, Ewan, take it away.
Great. So thank you very much for inviting me to give this keynote. As many of you know, I am a massive cheerleader for GA4GH, and it's just been such a pleasure actually being in the room and listening to the progress which you have all made sort of as the genomics community and as the standard setting community. It actually brings a real warmth to my heart about what we have achieved as a community. We have a long way to go. We've got lots more to do, but let's celebrate where we've got to as well. So, a little aspect for the people who don't know EMBL. We are an international treaty organization. We're based, we have 29 different countries which own us, which are in grey here, and we operate out of 6 sites. We're headquartered in beautiful Heidelberg, Germany, and our second biggest site outside of Heidelberg is EMBL EBI, just south of Cambridge, which does bioinformatics. Going to skip over this. Just to mention about the importance of data in this world and the way EBI works. Just a reminder that molecular biology, we have a long tradition of depositing data on publication. And what happens is that scientists generate data, they deposit at EMBL EBI or one of our sister organizations in America or Japan. We exchange data across the world, and then we transform, enrich, classify, and organize. And so the human genome and the human gene set, we are the master record keeper of that, and we are the master set of updating those genes and everything else. And we take that responsibility very seriously. Finally, we distribute this, both the raw data, so you don't have to trust us for the transform, but also the kind of transformed, enriched data, and give it out. And we get used worldwide, and please continue to use us. We're here to be used for this. Now, I just wanted to talk about the drivers behind AI, but give myself time for work that's come out of my own research group at the end. So I'm going to go fast. I apologize. So one big driver, and it's a big part of today, is genomics, sequencing DNA or RNA or nucleic acids. I will not take you through the technologies. They continue to develop. And it's interesting, as we've gone over the years, that our ability to sequence DNA has improved along many of these axes. And that goes from cost to turnaround time to error to whether you can see methylation to the number of molecules you can do simultaneously. An interesting aspect of this is there's no inherent trade-off of these axes. You're allowed to max out on all of these axes at the same time. And actually the genomics community still has a lot of technology headspace in being able to do that. So expect things to become longer, cheaper, quicker, less error-prone, the whole thing. That's the future here. That's enabled a whole bunch of things, and again, I won't have time to go into them, but in particular that integration into clinical practice, which we've heard so much about. I want to touch on the other big revolution that's happened over the last 30 years in biology, and that is imaging. We can now image at such incredible scale with such incredible field of view. It is remarkable. we can actually see molecules in vivo effectively or snap frozen at almost atomic scale. So this is just some examples from my colleagues across EMBL. I'll only do the left-hand side one here. This is from my colleague Julia Mohammed. This is a ribosome in bacteria. She snap froze bacteria. In one case provided antibiotics, in one case just the normal bacteria, and then could reconstruct the ribosomes in those two bacteria and see the direct impact of the antibiotic on the structure of the ribosome. This is the scale at which we can now do imaging. But both of these techniques, be it genomics or imaging, both require very high-dimensional analysis. And here we have had a real gift by our computer science and machine learning groups, colleagues, by artificial intelligence. Now, in artificial intelligence, it's really, at some level, something that I describe as calculus engineering, just like a massive linear optimization problem, just really amazing gradient descent. And I think for the people who come at it from a stats or maths background, the first time you look at these models, you're like, this is crazy talk. I mean, these are crazy numbers of parameters. How could we possibly believe that we can fit this? And it's taken quite a lot of confidence, I think, by the community to realize that you can fit these crazy big models. Of course, to do that fitting, you need a lot of really good data. So you're getting this combination of data engineering, really good data, such as we have at MLEBI, and really good calculus. And we can do all sorts of things, and I'd love to be able to talk you through some of the things we could do in imaging. Left-hand side is an MRI scan of a human heart. We've got other things. My very favorite sequencing technology, Nanopore, gets decoded using an AI system for its base levels. But those previous applications were things that I describe as a labeling problem, where you have to label the data that you're given. We also have AI that can do this also interpretation problem, which is I want to take this data set and I want to understand what generated it. What is the model behind this? And this is exemplified by AlphaFold, which trained multiple alignments to generate protein structures, and very convincingly has shown that it's able to do that regularly. And that blew a lot of us away who've been close to the protein structure field. This was a grand problem, grand challenge, stated in the 1970s. Many, many very, very clever people have tried to solve this problem. And it has been solved now, in effect, for these class of proteins in these ways. But what I'm going to come down to is now a piece of work from my own research group. It was our best bit of AI that I've been directly associated with. And this just came out in Nature, so we were all very smug about it a couple of weeks ago. And it made a lot of press, certainly in Europe as well, so it was very enjoyable. So this is work with, in particular, Maritz Gerstung's group in DKFZ, but also Søren Brunak in Denmark. And what we realized is that the language problem is quite similar to the health problem. So the large language models take a sentence, they tokenize the words. You can tokenize them in slightly different ways. And the goal is to predict the next word. We see a similar thing in healthcare, where you get a series of events in an order. Now, the tokens are not words, they're health events that are happening in someone's life. And the goal is to predict the next health event for that individual. Now, there is a big difference between words and healthcare in this perspective, and that is that healthcare has variable time. Variable time is just not a concept inside of large language models. It is an absolutely necessary concept inside of healthcare models. So if you have a heart attack and the very next day you are diagnosed with pneumonia, it is not a good sign. If you have a heart attack and the next healthcare event in your record 2 years later is pneumonia, it's fine. And that difference has to be represented. You have to be able to transmit that difference to the AI system. And so we were able to adapt large language models, or more formally, general pre-trained transformers with an attention network to handle this variable time. Those things are in red here. It's quite interesting about those adaptions, actually. And then we did the classic AI component in the UK. So we took a very large number of UK, the wonderful UK Biobank, 500,000 Brits, doing their wild British things. And we trained on 20%. We held back 20% of the individuals as a held back test set. We also held back a number of years as a test bed. Now that's very classic AI, but we also challenged it with a much harder system, which is we gave it almost 2 million Danish health records. We did not change the model at all. This is a zero-shot test, and we did not change the Danish healthcare records. It was exactly what was in the Danish healthcare records at the time. And again, we then tested on some held-back years. So in large language models, this was the prompt, 1936 to 2016, and then this here, the last 2 years, was our test. Now, I encourage you to read the paper. We think about this. One of the impressive things about this model is it naturally handles all diseases at once. So we handled all 1,225 ICD-10 codes at once, and it produces a prediction across all of those codes. These are 8 of those different codes. Here is heart attacks. The middle lines here, the population averages, and the points here in the light, the points are a downsampling of individuals from our model, where the output of the model is a rate of this particular event happening, and we express that as a rate per year. So a point here has a 1 in 100 rate of getting this particular code, the insulin-dependent diabetes, for example. The dark points are Delphi's, our model's scoring of people who the next token was the disease being listed. So the dark points are the kind of your positive set. So what you can see here is that the shapes of these curves are very different. Sometimes they're very different by sex. So this is breast cancer. These are women. These are men. Very different curves. Delphi is creating models that match those. The second thing is you can see the light dots lie above and below the line in quite an even way. That's what we expect. But the colored dots in general are above. And that is meaning that we are saying that these people had a much higher rate or risk of this disease happening than the population average. And so these points basically are the points which are we're predicting or we're able to predict the risk appropriately. Now, we compared this against a number of schemes, both schemes that were done in the clinic and schemes that have been published, including other AI schemes. I'm sorry to say there are a lot of pretty dreadful papers out there with some pretty dreadful schemes. So there were ones which were so bad we didn't even put them on the chart. Let's put it that way. But many of these ones here are things that are being used in the clinic, such as the QRISK-3. The Brits will know this. That's the method used for cardiovascular disease assessment. Or the Framingham, the Americans would know that one, for example. So here are 3 different codes: death, dementia, and CVD. Delphi is either the top or very, very near the top for across all of these things. There are many different ways of doing the assessment, by the way, and if you want to read those details, you can read them in the paper. Delphi not only produces good AUCs, so when we rank people by our risk, we're ranking them appropriately. We're putting the top risk people at the top and the low risk people at the bottom. But we can also convince ourselves that it produces a good probability distribution. So these are the rates, our model rate on the x-axis, and on the y-axis is our observed rate on hidden data for a variety of diseases separated by different years and sexes. And you can see that these lines lie on the x equals y axis pretty well. So our rates look realistic, basically. It's not only that our ranking is correct, but our rates are correct. And this is the AUC for the UK Biobank across all diseases. This is the classic AUC. This is kind of cheating because age and sex are such big drivers to health that you really need to factor that out. So this is it, the AUC versus an age-sex baseline control. And you can see that we are above an age-sex baseline control. Perhaps more interestingly was when we did this on the Danish data, exactly the same set of diseases. We don't change the model. We don't change a single parameter on a model, so it's a zero-shot test in AI speak, and we don't change a single thing in the Danish healthcare data, except that we take the intersection, so we only take the codes that are present both in the UK, codes which are present both in the UK and Denmark. And we do pretty well. If somebody would like to ask me about this, you can see the one place we don't do well is cancer. That is a very complicated story, which is a technical aspect, not, we think, a model aspect. Now, the exciting thing about AI is not only can it make these predictions, but I've got now really used to opening the kind of AI box and looking at the internal system of the AI. So AIs, you can treat them like a black box, but you can kind of also open the box and look inside. And this is us looking inside at the 1,250 whatever diseases. In this case, looking at the internal space that Delphi created for this. And this is looking at a UMAP projection of an 120-dimensional space, which is how Delphi represents this information. Each dot is a disease, and being near to each other means these diseases are close in that 120-dimensional space. Up here are cancers of the female reproductive system. Over here is diabetes, but also complications related to diabetes. Down here is the rather ominous death token, and a bunch of things such as septicemia and acute myocardial infarction in that space. Delphi knew none of this. Delphi, we didn't give it the hierarchy, we didn't give it any information about what the diseases meant, and it just created that space by the tasks being set to predict people's future healthcare events. And then very similar to large language models, we can sample into the future. So when you're doing this with a large language model, this is what you're doing with ChatGPT. You write a prompt, and then you get a sample of text that comes out generated by the model. So it's kind of going into the future from the text perspective. We can do that for people's healthcare. When we do that, there's lots of things to show that our sampling is looking pretty accurate in the sense that it is a good probability distribution of outcomes. So that's this plot here. And we get a kind of an increase over baseline of our sample trajectories, and that persists out to 20 years in the future when we do testing. Obviously, we're better in the first couple of years than we are at the end. Now, we can now bring just like the full techniques of AI and generative pre-trained transformers to this problem. And there is a whole community and a whole system coming up with numerous innovations in this field, and we can just lift this now across into epidemiology. So we are making hay in the sunshine at the moment between the different research groups behind this. So we can put in environmental measures. We were thinking about PM2.5 and PFOS, for example, but anything. That's a kind of token-based system. We can put in drug prescriptions. Again, we're doing that at the moment. We can put in genetics. We can put in imaging. And we can put in metabolomics. And when, by the way, for imaging and metabolomics and for genetics, we have to think about the representation of those things for the network to get hold of. But again, this is rather similar to the text and image fusions that are happening inside of AI. So these businesses where you type in some text and it produces a video for you, that is the same sort of fundamental technology that we're leveraging here. So with that, thank you very much, and I'm very happy to take questions.
Yeah, thank you very much, Ewan. I think this was a really great talk. I think it's fair to say that your model combines a lot of individual tests now that clinicians typically apply. How can you see in the future that we could use your model in the hands of policymakers or stakeholders?
Yeah.
How could they benefit from it as fast as possible?
Yeah, I think it's going to be interesting to think about. So our model, I think, is very flexible for research, by the way, and I think there's lots of research directions we can take it. We think a very easy part of implementation is actually doing healthcare system planning. And that is because we can handle trajectories out in the future. We can take all of, I don't know, Uppsala, and then model 1, 2, 3 years in the future about what we expect the healthcare events to happen in this. And we're relatively confident that that model is better than a simple age-sex baseline. Many people currently use an age-sex model, demographic model, and we could do better than that. And what's good about that is it doesn't require so much sort of regulation. Thinking about this in decision support as well for clinicians, as you know, Q-Risk and Framingham is used at the moment. It's obviously feasible there, but notice that will require, so you definitely wouldn't use this model because it's trained on UK Biobank. And so, for example, our model refuses to let anybody die before the age of 40 because it's only been trained on people that were recruited to be healthy in the ages of 40 or 60. So our model is not appropriate for rolling out inside of a healthcare system straight up. You'd have to retrain it. But you also need to think about the intervention side as well, of course, and that's on the clinician side, not on the risk modeling side.
Okay. Can you comment a little bit on the distribution of the model? Basically, who should have the weights? Who should get the output?
Yeah. As a research tool, we've published it. The code's on GitHub. You can download it. With discussion with UK Biobank, we've decided, for an abundance of caution, the weights are considered personalized data at the moment. So you have to apply through UK Biobank to get access to the weights. We think that's appropriate. You can retrain it, of course, on your own. You have it. Yeah, it's you know, you need a reasonable GPU farm. It's not massive. So yeah, we think there's lots of optionality there. Of course, it's again, if anybody's going to use it in a clinical setting, it would have to go through regulation. I mean, clearly, just as other, you know, just as Framingham and Q-Risk and other screening risk assessment techniques are done at the moment.
Okay, great. Can you comment a little bit on how much more data would benefit, or maybe I should rephrase that. So, do you have a trajectory of more data means more accuracy, or? Yeah,
I mean, it's very clear. You know, never ask a statistician how much data you should have. You know,
That's right.
It's always infinite. So yeah, clearly, very clearly, and you know, healthcare does have a lot of rare events. And when you think about combinations, say of genetics and disease or rare events, you know, you've got the thing multiplied. We don't think the model is robust once you see there's about 1,000 events in your training set, and it stops really working when it's below 100. So somewhere between 100 to 1,000 events. Of course, if the event is 10%, then you don't need that many people. But most healthcare events are at the 1%, 0.5%, and interactions, of course, far lower. So I reckon we will get better and better results. You know, 10 million, 100 million. That would be the place I'd like to go. 10 million first. Would be a good number.
Oh, exciting times ahead.
Yeah, don't forget Finland's got 5 million people. People can do that, and we could do that in Finland. Denmark's got 5 million people. The Danish data set is about 10 million strong because they've had it for so long. So yeah, that's not crazy. Those aren't crazy numbers.
Okay. Yeah, maybe let's switch to Slido. Yeah. I think a few questions have come in yet. First question: can you include costs and resource use? So can you comment on that a little bit?
Yeah, it's not. It's fine. You know, you need a modest GPU farm. Modest. I mean, you don't need to go crazy. I don't know. Maybe I'm not against us going crazy and seeing what we can do, but I think we're data limited, not GPU limited at the moment. Okay.
All right.
Is that? Do you think it's? Yeah, I mean, definitely. Right, if it's definitely going to be used inside clinical decision making, it's got to go through a process for. I mean, it's got to go through a fully worked out process for interventions. By the way, I see this very similar. I had the privilege of being a non-executive director of Genomics to England. I still am. You know, watching that transfer from a research, being confident about research to being delivering inside of a healthcare system, a lot of it has got nothing to do with can I generate the data, can I model the data, can I understand the data? It's how am I going to implement it? What's the consequence on my healthcare system and that sort of thing. I see this AI stuff in a very similar way, that we're right at the start. We're a bit like Genomics was in 2010, 2011. And we're going to have to roll it through for the clinic.
Right. Next question is about cancer.
Yes.
I think you mentioned it briefly.
Yeah.
So why was it more difficult?
Yeah. What's going on?
Yeah.
So, gosh, So I've learned so much about AI, but this is actually a classic epidemiological problem. It's complicated. So, UK Biobank was recruited between the ages of 40 and 60 in a particular time window. Actually, the kind of modal age is sort of 45, 50 to 60. That's the modal recruitment or something like that. The UK Biobank cohort is hitting kind of peak cancer incidence now. And then you have to add the fact that cancer diagnoses, because of the data stream that they come through in UK Biobank, they sometimes arrive later than other tokens. And so actually, if you look in UK Biobank, we have this exponential rise of cancer diagnoses, and then it flattens off very counterintuitively. And we don't think that's anything to do with the natural history of cancer.
It's a labeling problem.
It's just a sampling frame problem, yeah. Now, in Denmark, we're not sure about whether they have the same delays. I actually think they do. But the key thing about the Danish data is it doesn't have that modal recruitment time. And so the Danish data doesn't show this age shift. So we think this problem is going to solve itself with a better ascertained data set. It reminds everybody that you have to ascertain your data really, really well. Yeah, that's an incredibly important part of running these models, is well ascertained data. So knowing what your ascertainment is.
Okay. Next question, a bit longer. Thinking about the marginal return on data, if all the cohorts and project represented in this room made their data easily accessible for training a single pooled Delphi-like model, so all the training data that you can possibly think of, basically.
Yeah.
How do you think that would improve the model's performance? I think we just
it will definitely improve it. I think it's going to be a very interesting question. To what extent do we use separated data sets as challenge versus, increased depth? You can think of healthcare systems. The AI community has this concept of style or theme. So, please rewrite this letter in a formal style. And so you can think of healthcare systems of having a style. And you can learn the UK style and the Danish style and the Finnish style. So there's quite a lot of tricks we can play here. We don't know until we've done it. All I can say is as long as we do it right, it won't go down. Fingers crossed.
Yeah. All right, next question. How will AI affect genomic standards already out there and yet to be developed? What is needed and how will our current tools break?
I don't think AI is very data needy, and it's very needy of well-structured computable data, as everybody knows. That has been the standards mantra and the bioinformatics mantra way before AI. Structure your data, deposit it with the correct metadata, do all of these things correctly. So in many ways, our previous practices about standards, be it in research or in GA4GH, in this research to healthcare zone, has been the right attitude. I'm sure AI will illustrate some areas where we need to do better in our metadata. But I think it's quite hard to predict where. And then AI can be part of the solution, in particular kind of lab in the loop where they can, thinking about automated ways of upgrading the metadata by reading papers and then maybe handing it to a human to check. You can see ways of AI, that's already happening in the EBI for improving the way our throughput of metadata capture, metadata or curation. So there's a lot of opportunity. There's as much opportunity as there is challenge with AI coming into this space.
Okay. I think we are out of time for the questions. So thank you again very much. Round of applause. And And our next speaker is Mathias Ekman. He is the Director for Digital Health Platforms and Partner Ecosystems at Microsoft. He's also part of the governance board of SweLife and the advisory board of Genomic Medicine Sweden Informatics. And last but not least, he also holds a PhD in theoretical physics, focusing on large-scale computing for solving problems explained by quantum physics. Please, Mathias, stage is yours. Yeah.
Thank you. And on the last topic, you know, I never thought when I left the not slow-moving research world in quantum physics and changed to working with healthcare system and healthcare providers that we should be in this like intersection between quantum and data that we are now. So it's extremely fascinating. I will spend a few minutes here talking about some of the trends and building on the great presentation that Evan previously did. And I think that one of the main challenge, I think, in the precision medicine and the society is this too big, you know, loops. And you mentioned the lab in the loop at the end, that we have this like drug discovery research loop, and then we have this people on patient are passing through the system, and we have the clinical loop. And one of the challenge is that these loops are like measured in years. Of course, from a drug development, we all know that it can take huge number of years and not everything will succeed. But the same is true, sadly, for the clinical loop. Update of best practices to ensure that new findings are scaled to more than 80% of clinical people. One can argue if it takes like 15 years or 10 years or 5 years. But these loops, sadly, is in years. And then we have the challenge of translating world-leading research from the left-hand side into the other one. And the question is there a change now with all these like large language models and generative AI that fundamentally change how this can do and reducing the time? And I should say yes to that question. Previously, I should say that a lot of the technology that has come is solving a point problem. Maybe we need to integrate data from the EMR to the research side. It's something that is happening in one step. But as mentioned, since there is so many activities in these loops that are manual, or you need to explore, find information, you need to gather real-world data in the real-world data study, these models has the possibility to remove a set of these like manual steps or these things. So I should say that start to think about these models. That is something that is trying to solve things in different steps. It's not one thing or one solution. And I have had the pleasure to work with several organizations from the pharma side, UCB from Belgium, how we look in entering generative AI into the drug discovery loop. Another good example is around rare diseases in Spain, how they are like Foundation 29, an organization has built a chat-based system that they handing to primary care to ensure that in primary care there is an idea of trying to better find people that are undiagnosed. And then, of course, you have previously mentioned the work with Brainchild that is funded by the Childhood Cancer Foundation in Sweden together with GMS. I will double-click a little bit on that. But for all of these projects, there is a component that these new type of models try to solve something. And in fact, we are entering an area where we have multimodal biomedical AI. I think you mentioned images. Images plus language has given us the opportunity to start to chat towards images. And what we see around that type of development is that we're going from treating this large language model as more like fundamental platform capabilities, that they become trained on specific things. One such example is the work between Microsoft Research and Providence in US that built one of the largest fundamental models for pathology. So very domain-specific fundamental model that you can start to use and introduce. And we see that this is possible across all of the image modalities, that you basically combine the large language models that has been trained on the internet, but has learned how to reason, but then in the same way that they were like able to modify this type of model to enter this like timeline concept. You take this large language model and contain it to say you should be an expert on this specific content, for example, pathology images. And we had these type of studies of trying to predict patient pathways. And the last thing is also this idea of trying to help the clinical people of making sense of data or even understanding the process. And this is a recent collaboration around rare diseases to give clinical people some kind of AI assistant that helps with things. But the fascinating thing is that proteins are language too. It's a set of letters, and in the same way that you argued around healthcare trajectory, of course you can start to guess, but now we're not guessing the next letter in the sequence. We try to guess what the function is, not the structure, really the function. So there's a lot of work going on currently to try to use the same type of training techniques that we have done for the large language models to try to train on the sequence to understand what that protein in fact has to do in a specific solution. So just a set of research paper that I can share later on, but I think that the combination then between trying to understand the protein structure, the image, as well as the clinical nodes, gives the opportunity to try to rethink how we can better provide care for patients. And this is the task for this brainchild project that I know that you heard about. And I think that the fascinating things is, and I will show the video shortly with David in it, but the human brain cannot cope with all these type of data. But could we, based on the spirit in Sweden, try to gather a data set or a structure for exploration where the goal is very, very challenging to have 100% survival rates for children with cancer?
There are two grand challenges. One is to make all the children with cancer survive. We are now up to 85% survival in Sweden, which is really, really high. The second challenge is side effects from the heavy treatment given today. Project Brainchild is a data platform for childhood cancer patients. We take big data types such as whole genome sequencing, imaging from pathology, imaging from radiology into one data solution, the Brainchild project, using Microsoft's platform. It's so much data that the human brain can't cope, so we want to leverage the power of machine intelligence. It's of course a way of finding new patterns that will lead us to better drugs. What we want to do is to use the Swedish spirit of innovation and bring these data types together into one integrated solution for exploration. The hypothesis here is that if you use machine learning to explore the data, we can start a new evolution in childhood cancer towards a 100% survival.
And I think, to close, what is worth to think about, that this concept of what we experience when ChatGPT arises and these type of large language models, this is the first time we have never had this like broad type of general purpose technology that has spread across the world so fast. And it has spread as a more as a platform concept. And I think that starting to think about how we will be able to leverage that type of fast scaling when it comes to these like research cycles, and ensuring that the cutting-edge world research that is done in part of the world can scale to the broader world. I think that is the end game, at least for me. So thank you for listening.
Thank you, Mathias. Really inspiring talk, and yeah, good times ahead, I think. Yeah, our next speaker is Joep de Ligt. He holds a PhD in molecular life science with quite an impressive publication record, and he is the data lead at Hartwig Medical Foundation in the Netherlands. He has led and advised national and international genomic efforts in human and pathogen genomics, and he was the driving force behind the genomics effort to sequence more than 80% of the cases in the COVID-19 response in Aotearoa in New Zealand. Joep, yeah, please take it away.
Thank you. So after these great talks about some of the really large initiatives, I'll try and take us down a little bit more towards the day-to-day reality and what we're trying to do in the Netherlands. So we don't have a national initiative that is government mandated. We have some local initiatives that try to be that. So we're in adult cancers, also people from the Princess Maxima in pediatric cancers are here today. But we really like to think in this learning healthcare cycle. And I was thinking, I've seen about 10 different versions of these last 2 days. Maybe we should standardize this one as well. But we really focus on two things. How does this actually get to the patient? So in the Netherlands, there wasn't a service to do whole genome sequencing, so we made one. So now Dutch patients can get whole genome sequencing. Some of that is actually in routine care. And at the same time, we always ask those patients for consent for reuse. So we do not do research ourselves, but we hold this data. And we try and make it as available as possible. So there were some questions earlier today and also yesterday. We've actually shared this with over 300 different research groups, including Singapore, Australia, Korea. So it is possible, even under GDPR, to share your data with those people and have them use it. Just so that you know. But then also to try and take those innovations then back into the clinic. And that's both on the data side. So we've spent a lot of time on optimizing compute so that it's now only $12 to analyze a whole genome for tumor in a cloud setting. But also on the sequencing side, that we're innovating on the sequencing technologies. We're testing Ultima and Roche technologies in Europe to see if we can bring that cost down. Because the person that made the comment about the cost of whole genome sequencing is not the point. I would love to have talked to the Dutch agencies that deal with healthcare insurance, because they think differently. So one of the things that we saw a large unmet need were cancers of unknown primary. These are patients that have a very poor outlook in terms of the available treatments they have. There's not a great survival for them. And there's no standard of care because the clinicians don't know where the tumor has come from, what tissue it is, what sort of the types of treatments are that could work. And the other thing is that they typically go through a very long diagnostic odyssey. So if you have only expected lifetime span of about 12 months and half of that is taken up by diagnosis, that is not what you want. So we thought, now we have a data set of these cancers that we've seen from the Netherlands. So at that point of time, we had about 4,000 patients, and we thought, can we use machine learning to learn how to predict the tumor and the tissue of origin? So we used over 500 different genomic features. So this is not taken into account clinical features, just the genomics, tumor mutational load, MSI, all of those features. And we tried to predict the tissue of origin for 29 different cancer types. You might have seen publications that do way more. But these are the clinically actionable ones. So that's what we focused on, because they actually meant that they could give a different treatment to these patients. And it had about an 80% accuracy. Again, you could probably get higher, but this is what was actually useful and understandable by the clinicians. They understood which features were going into it, what it meant. So that was implemented in routine care in the Netherlands in 2021. That was largely driven also by patient advocates that were seeing these poor outcomes. And what we could show is that in about 63%, we could provide a information that was useful to the clinician to determine the actual tissue of origin. And that there's another 11 and 6% where there were additional findings, either targeted treatment options or germline findings that were considered clinically relevant. So in about 67% of these cases, there was a care option provided that was otherwise not present. Unfortunately, not all of these patients start on this treatment because some of them, their health just doesn't allow that. But we saw that the half of them that did start on it did survive significantly better than those that did not get those types of biomarkers. Now, the other thing that clinicians tell us is that it's very hard to curate and structure the clinical data to use in their decision-making processes. So that's another project that we have where we use large language models to assist in the structuring and curation of what we consider total free text forms. So between hospitals, there's still a lot of these amnesis or sort of clinical reports that go from one hospital to the next. So this is a system that we've now made, and this is really focused on the clinical data. So there's very minimal or no genomic data in here, but it really tries to read those clinical reports and extract those terms that have been mapped as to be relevant for this patient's outcome. And to then also have a curation model behind that, because of course these models are not yet perfect. You can constrain them with the types of terms that you're looking for, but we still for now want a human in the middle because we want this to go out to the clinic. So here's one example of a molecular report and how that then structures it. So on the left is sort of a template on what a typical medical referral letter can look like. But then it finds these terms somewhere hidden in the text, and they might be all over the place. But it basically says, well, there is an event in FOXP1, and then it translates that into a genomic model that can then be curated, like did the model make the right association here, and is that what we actually can use? Now, if you can do that, and you can systematically mine these medical records, because in the Netherlands they are not standardized at all, you can do some really interesting things. So the first thing you can do is you can do better information sharing with the actual expected outcomes. So here on top you see a couple of different treatment options, and you see the data as it was obtained in the clinical trial. But then also a matched cohort with real-world data. So we try to match it on as much of the clinical parameters as we can, and then we look in the Dutch Cancer Registry for what those patients, how they actually survived and how they actually responded. And we all know that real-world data is different from clinical trial data, but this really, in the report, makes it available to the clinician what it really means for this particular patient to be treated with this type of drug, seeing their history. And the other thing is that we can do is we can do better trial matching because we now have a standardized readout of their clinical parameters as well as their genomics. We can recommend the treatment. And the last thing I have for you is a challenge because there's things we can already do. So this is a fairly simple linear regression we did across our entire database with clinical durable benefit as an outcome. And these are genetic biomarkers where there is a very strong non-response. These are patients that are being treated with drugs that give side effects, that cost money, and that are not having a benefit, at least with our, for the moment, fairly loose statistical criteria. And that's because the numbers are incredibly small. We have a data set of slightly over 7,000 patients, but once you get to a certain event with a certain treatments, groups become very small. And one example here that I've highlighted is in skin melanoma patients, there's immune therapy, and this is mostly driven by B2M deletion or double hits, where there's not a single responder in our group. And if you look at the Kaplan-Meier curve, you can appreciate that this group is the group that does not do well. But there is still a gap to be closed, because if we want this to be clinically actionable, we feel that we need to give a certain that this patient is indeed not going to benefit. And the current selection that we have here is on the left. That's the 166 patients that we have in the database with this treatment. If we want to be certain that it's less than 5% response, we need almost 1,000 patients. If we want that to be less than 2%, that needs to be about 2,500 patients. And within the Netherlands, we're not going to get this. So we need you guys. And this is not AI, this is simple logistic regression. And anyone that has data on melanoma patients treated with immune therapy, let me know the statistics, what you're getting, because we love to know if this replicates across other cohorts, and if we could get to the number to make a claim like you shouldn't treat these patients if they have a deep B2M deletion with immune therapy. The other thing is, if for this group of patients that we've now tested, if you would have withheld the treatment for these 11 patients that we think would not respond, you would have paid for the sequencing of all melanoma cases. So also the socioeconomics of this, they work out, because you can actually make a profit. There's more money left than you would have spent on treatment that doesn't work. And with that, I thank you for your attention.
Yes, thank you, Joep. This was a really nice presentation, very insightful and very close to our daily life, I guess. Yeah. Our next speaker is Nevenka Dimitrova. Nevenka currently is a computational oncology advisor for Memorial Sloan Kettering Cancer Center. She holds a PhD in computer science and has an extensive portfolio of over 140 publications and 80 patents spanning healthcare, life sciences, and technology. Her career spans scientific leadership roles at Philips, Glaxos, GSK, and she contributed or even founded many other companies and contributes into the institutions around the world. Enjoy.
Thank you. And for me, it's an incredible honor and pleasure to be here. I learned so much in the past 2 days. And especially all of these country-wide initiatives, it's really amazing to come back to GA4GH. I think I've been here like 10, 12 years ago when it started. And it's back then we were just dreaming about the wealth of data that we have today. So with that, I'll start and say currently I'm at Memorial Sloan Kettering Cancer Center, and I have several themes today to talk about. As usual, I'm very excited about what I'm doing, and I'm going to talk about things that are from multi-omics to spatial technologies. And they're progressively more complex and more insightful, not necessarily the insights we get are more clear, there are more questions as we discover more. And my current fascinations, my current project is really on spatial biology, both spatial proteomics and spatial lipidomics. And I'm also involved in AI and federated learning hackathon. I will devote a little bit of time on that. And then I want to talk about GA4GH, going from sharing data to sharing models. Ewan alluded to that, that it seems like even sharing the weights of a model, it seems you need to do it in a secure and private manner. We currently don't have any guidance on that. People just put out there their models. I'll talk a little bit about traversing the scales. The Zuckerman Research Center is where I'm currently. There are 120 labs, 600 postdocs, another 300 trainees, and thousands of oncologists, radiation oncologists, pathologists, clinicians, and so on. Some of these initiatives you're aware of, like CBio portal, started actually at MSK. Part of the data is also in Cancer Data Science Initiative portal, which is internal, but part of that data also, the MSK impact is in a CR Genie project, which is really incredible. And last year, there was a foundation model for computational pathology published along with a few other institutions. Millions of slides went into this model, and that is now at the service of everyone else out there in the clinic. And the CBio portal, how many people here have used it? It's almost half the audience, and it's growing. And what is incredible is the sheer resource and all of the data sets and how many publications have been derived and how many publications have been helped with this resource. So with that, I want to talk about our current initiative, which is called Cancer Ecosystems. And after all of these years of studying mutations, having genetic tests with 500 genes, and also studying whole exomes and whole genomes, whole transcriptomes of cancer patients, now for the first time I feel we have eyes, we have a zoom in into cancer biology. Why? Because this technology, I'm showing a few slides which are zoomed in version from CellDive, from Leica. In the same slide, we can see at the top structural markers such as PAN-CK, which is an epithelial tumor marker, Ki-67, which is proliferative marker, and other markers, CD31 endothelium. Lymphoid markers, which is the adaptive immune system. You recognize FOXP3 because it's all over the news. The Nobel Prize winners for physiology and medicine were announced yesterday for T regulatory cells, and then myeloid, which is the innate immune system. So neutrophils, macrophages, and platelets. So what you see vertically is different samples. From untreated sample, it's from the same section, but these are three different views. So we are looking at different layers of cancer. One is structural, and also how the immune system cells are with respect to these tumor cells. And then how this progresses under treatment. So this is under immune modulator, this is radiation treatment, and this is combination of immune modulation and radiation. And as you can see, the cancer cells disappeared. Now we have more or less structurally good-looking tissue, but also quite a few activated lymphoid cells, as well as myeloid response. This is from my colleague, Paul Romeser. Together, the two labs, we have over 100 slides. I just want to tell you that this new technology is a repeated staining. So you overlay one antibody, you wash it away, then you overlay the next antibody, and so on. So it's possible to have 60 markers on top of each other. And for the first time, we can see what is the physical interaction between these markers and go and dive deeper. So in research, what we are doing is we are using spatial technologies, as well as we're hoping to use microbiome and create foundation models, AI foundation models to detect, classify, quantify the abundance of these markers of disease, and develop further understanding of ecosystems. What you see here is a vision transformer, but also we're hoping to use federated learning and transfer learning, so we can transfer everything we learn on the research samples back to the clinic on H&E stained images. So the goal really is to have resolution agnostic understanding of tissue classification using zero-shot, not zero-shot, class prototypes, as well as infer new phenotypes. And this is especially true when you're looking at pre-malignant milieu of cells, because for certain markers, we don't even know that they're actually the pre-malignant markers until in-depth studies are performed. But also discover new biomarker models, new drug targets, stratification of patients. Carolina this morning described that there is a protocol to stratify AML patients into 5 subgroups. And with these kinds of technologies, we should be able to do further stratification of the patients and have individual prediction of response trajectories, just like what Ewan showed for different diseases, but now this is in the course of the disease. This is the work so far. We are basically decomposing these images because each image is something like 8,000 pixels by 120,000 pixels, 500 gigabytes worth of data. You can't even open it on a regular computer. You can't definitely analyze it without GPUs. And then we have a flexible taxonomy that we can change, and based on the markers and based on the cell phenotypes in the taxonomy, now we can do image preprocessing, remove the parts of the tissue like here. We are not going to load this into the vision transformer model, and then we can perform learning, meaning we can continue to train the model, into, we call it second foundation model, because the first foundation model really is Dino. In our case, it was V2, now it's Dino V3. That's for Facebook research. Facebook spends about $20 billion worth of funding every year on AI research. Some of this is really computational resources. For example, to train Dino V3, they spent 60,000 GPU hours. Now, what I'm going to show in the next slide has very limited GPU hours, and I'm hoping we can get more help and more support through federated learning. But just to tell you that when we say foundation models, this is what it takes to develop a foundation model. So what we are able to do with this is to take this tissue and perform clustering based on these neighborhoods and cell phenotypes of neighborhoods. And then when we overlay the markers, then we can see the visual representation. In this case, it's UPAR. UPAR is, if you remember, one of the most ubiquitous biomarkers for cancer. And then assess diversity. This is basic Shannon entropy, but we can compute it as diversity of clusters, transition zones between tumor and immune microenvironment, and so on. This is just 300,000 images, 1 week of NVIDIA H100s, 4 GPUs, and what you see in this slide, and I'm very glad that we're able to produce this, is all of these lineages from the endothelial, epithelial, and you can basically see them. There are only a few dots that are in the wrong place, but again, remember, we are training on one slide, slide 58. And now this is evaluation on a completely different slide. And it can already almost reproduce the lineages, which to me is really mind-blowing. Another aspect that I wanted to talk about, and I'm really surprised that at this conference, very few people talked about, which is microbiome. We have an ongoing hackathon on microbiome data. The microbiome data is from Snyder Lab, from Stanford. And we are using MPEG Genomics compression standard, which is actually a standard not only for compression, but random access, security, privacy, and provenance of data. And I'd like to give a shout out to a few individuals, I think they're here in the audience. So Jaime Delgado, I don't know where Jaime is, there is Jaime, and Patrick Chung, hi. They've been involved both in GA4GH, but also in ISO MPEG Genomics. And it turns out that even undergraduates in this competition can take this type of compressed data and perform AI analysis. And the goal really is to build a machine learning model that can classify microbiome data. So you have raw sequences. Can you classify the body site that it came from? And this is just the data in the hackathon. One of the data sets is from Stanford, the other one is from Viome. And this is mostly people with type 2 diabetes. There are 5,400 samples, but these are 86 participants over 6 years. Now, the key finding was that the microbiome is really a fluid systemic ecosystem. And, 1 minute. The point is that we were able, these participants were able to have greater than 99.6% accuracy, so going from raw sequencing data to correct classification. And the next track is really ongoing, so you can join. Here is the link. And the next track is how do we associate microbes with human cytokine expression, meaning how do microbes and the human immune system talk to each other. One of the solutions has federated learning. Federated learning was a separate track. And the federated cyclic model. So you train a local model, you pass it on to the next site, that passes it on to next site, and so on. And this is totally doable if we just had some kind of a framework to exchange these models. And I know I have less than 1 minute left. But what would be really nice is if we're able to traverse the scales, that in the images, both on a macro level, radiology, pathology, there is very rich phenotypic information that we can associate that with everything that's, say, in spatial transcriptomics, mutations, spatial proteomics, and this is really a picture from Rosetta Fold, which was David Baker's work, and last year he got the Nobel Prize for his work on predicting protein folding. So what I want to leave you with is this picture that we can go in increased molecular depth from genomics to multiomics to spatial ecosystems, but also we need to expand the human ecosystems, which is from single node to a global network. And here is where Global Alliance can play a role, where we can not only break data silos and human silos, which sometimes are more difficult to break than the data silos, and we can benefit from equity, diversity, reproducibility of the data, ethical and global collaboration. And we can work on shared discovery, diverse and ethical innovation. And this is because some of this data are very rich but very expensive. Just one of the experiments that I showed you can be anywhere from $6,000 to $10,000. This is like genome sequencing in 2009 or 2010. And now we have the genomic data, but we need to garner other types of data. So this is why here is my call for engagement to have in the federated AI work stream to have a new project that connects multiple medical centers so that we can aggregate these spatial proteomic data along with, so we can learn from spatial data, predict genomic and the other way around, and then translate hopefully to the clinic. Thank you.
Okay, thank you, Nevenka. Again, a really inspiring talk, and I think it shows how we are quickly entering a world where AI is without alternative almost to model all these different types of data. Our last speaker is Man Zawati. Man is an associate professor at McGill University's Faculty of Medicine and Health Sciences and a research director of the Center of Genomics and Policy in the Department of Human Genetics. He's also an associate member in the Department of Medicine, the Department of Equity, Ethics and Policy, and the Faculty of Law. And last but not least, he also co-leads the regulatory and ethics work stream here at GA4GH. Man.
All right. It's a pleasure to be here. I know that we're almost at the end of the session, so I'll try to be actually briefer than 10 minutes to give us a bit of time for our Q&A, but it's wonderful to be here, and thank you for the kind invitation. So as I was listening to this conversation, and as I was also asked to talk a little bit about the normative landscape of AI and the work that we do at the regulatory and ethics work stream, it kind of came to my mind in terms of convergence of AI, genomics, and law, or humanities just more generally. It reminded me of a recent workshop that we had where we were talking about how AI today, at least in terms of the discussion, is to data was what genomics was to biology when it first emerged. And here we're talking about abundance of data. We're talking about issues related to equity, bias, and of course, talking about the ethical and legal challenges that are associated with this. Now, dealing with ethical and legal challenges is overwhelming, but rest assured, the lawyers are here. I only have 8 minutes, so I want to talk to you about the key updates on AI, the normative landscape. I want to talk to you about some of the key normative approaches related to AI, and then talk about some of the key next steps towards responsible use of generative AI and some of the work that we're doing at RUSE. So, this is definitely not an exhaustive list, but just to give you a few examples, there are 2 jurisdictions worldwide that have adopted a very comprehensive AI legislation. You've heard about the European Union Artificial Intelligence Act, but there's also South Korea with the Basic Act on the Development of Artificial Intelligence, and I like this part in their title, the Establishment of Trust. There's a few jurisdictions that have announced and release similar draft bills. Many of them are in Latin America, Brazil, Colombia, Chile, Peru, Argentina, Mexico, but also in Taiwan and in Kenya. I left the Canadian one here for Canadian colleagues. There was a bill, and it's a very comprehensive bill on artificial intelligence, but for political reasons, before the last election, the parliament was prorogated. And what that does is, wherever you are at in the stages of your bill, it kind of dies off, unfortunately. But there's also many jurisdictions that have adopted sector-specific regulation or norms that apply to specific uses or devices involving AI. So if you take the example in Quebec, we have some new privacy legislation that touch on some of those aspects. In Hong Kong, personal privacy ordinance in the US. And then you have also, of course, specific to healthcare or AI-enabled medical devices. So Health Canada is a good example of that, some of its pre-market guidance for machine learning-enabled medical devices. And then you have also this approach of focusing on a particular type of AI. So in this case, generative AI. This has happened in China. And also another example of this is Saudi Arabia with its generative AI guidelines. AI has been really out there in the last few years, at least in the news. But from 2017 to 2025, we've had 77 countries that have launched their own national AI strategies. Most of them happened in 2019. There is a lot of soft law tools as well. So this includes ethical guidelines or what we call voluntary frameworks in the UK, and Australia, and Singapore, and Thailand. But one thing that we've noticed as well is the fact that this has also increased international and regional cooperation, whether it's in Europe or whether it's in Asia with the US, but also in the Middle East and the creation of the Arab Dialogue Circle on Artificial Intelligence. So there are a variety of different key normative approaches. The one that we've probably are most used to when it comes to regulating science is the risk-based approach. In South Korea, they talk about impact, so high impact AI. But these are, tailored to the different levels of risks. There is potentially less stifling of AI innovation. There's reduces business costs, but it's also very challenging and foreseeing those potential risks, and also in defining them. But you also have a principle-based approach, and usually you find those in ethical guidelines, where you would see the general principles around promoting transparency, ensuring human oversight, respecting privacy. One of the great advantages of that is, of course, agreement on those foundational values. It does provide greater flexibility. You don't have to go through multiple loops to be able to change those guidance documents as if it was a legislation. But there are also difficulties when it comes to interpretations and enforcement. And there's also the self-regulation approach. There is more flexibility here throughout the life cycle of the AIs, especially if you want to modify or update the rules. It's always possible. It is expertise informed or expert informed. But again, here some of the issues are with compliance or enforcement. And here at GA4GH, the Regulatory and Ethics Workstream has been working on guidance points for the responsible use of conversational/generative AI. And what I want to do in the next very short few minutes is just to give you a snapshot. Now, this is also an invitation for those of you, hopefully, who are staying with us for the Connect sessions. I highly recommend that you join us tomorrow for the Agentic Science session, where you'll hear a little bit more about that, but also what GA4GH is doing related to AI. One of the general principles is, of course, about human oversight and epistemic authority. The approach that we wanted to take is to identify where the challenges are, but then work with colleagues about how we're going to actually be able to implement them, right? So there are issues or challenges with human biases, whether it's automation bias or aversion bias, some of the limited knowledge, especially if you're querying those genomic data sets. But what we really want to be testing, and this is the next phase of our work, is really on implementing those transparency requirements and provide guidance on how to respond to those potential issues, including methods to mitigate biases, perhaps even at the query level, and increase awareness of the system's limitations. And of course, determine key moments for oversight and identify possible human interventions. This requires a dialogue with, experts in ethics and law, but also, of course, our developers and folks in computer science. Questions around safety and accuracy is another example. This is related to identification of hallucinations. And here, some of the guidance points that we want to test is how to train users on identifying those inaccurate responses, how to facilitate incidents reporting, but also provide mechanisms for the filing and handling of complaints. So again, this is just a snapshot. I'm almost out of time, but I will stop there to give us a bit of time for our Q&A. Thank you very much for your attention.
Yeah, thank you very much, man. Please take a seat.
Yeah, thank you.
And I may also ask the rest of the speakers to come back. All right. Well, we have our first questions already. First question. How do you hope the models that we have heard about just now will expand into other agnostic areas on a large-scale basis? Anybody want to go first? Maybe that's a question for
Yeah, I don't know
Ian.
what agnostic areas. I just think bring it on. It's honestly AI. I was pretty skeptical 10 years ago. I thought this business of way too many parameters, many more parameters than data points was like a big no-no for doing robust statistics. But I've got really used to it, and I've definitely transitioned not only into having kind of about 5 years ago I had grudging respect for AI models, but I still didn't like them. And now I really love them. So I encourage people to sort of dive in, because I think they're really flexible ways of handling. Basically, a lot of biology is very non-linear. We have a lot of very non-linear spaces in biology. And that's always been a challenge for how we've thought about analyzing data. And we've now got a toolkit which allows to, if you can think about it, can learn these non-linear spaces far better and in a far more rich and complete way.
I mean, to me, agnostic is also with respect to any type of human tissue. Many of the methods that are for, say, computational pathology, you can apply them to cancer, you can apply them to fibrosis and so on. So it's not necessarily disease specific. So I think many of the methods are transferable. Some might be, we might do some refinement and tuning, and then it's just amazing how the results look like. To me, the turning point for me personally for neural networks was when they taught a neural network how to play a video game. At that point, I said, wow, this really cannot be any longer ignored. I tried probabilistic neural networks 20 years ago for face recognition, and I was debating whether to publish that paper or not. Eventually we did. But with a lot of data, and if we're able to federate this data, and federate the learning of these models, I think we can do a lot together.
And also, I think if you look at your research of this, like predicting the future, and thanks to the construct of the models that you basically can talk to that like model, also gives an opportunity to expose in the future that to individuals, so that they can start to try to understand of the different pathway that I may enter. Mm-hmm. What could I do to make a nicer path and avoiding things? So I think the combination of these models of like predicting, but also helping people to understand the decision that one needs to take to avoid.
I do think there's a caution here from the epidemiology side, which is the models can be predictive, but I don't believe the models are causal. So that counterfactual, what could I do? What happens if? That's a different model than straightforward prediction. And I think we have to be very careful about separating the kind of very often we want to ask these counterfactual questions. This is my trajectory. What could I do to change my trajectory? And that's not quite as simple as that's not as
you cannot just revert it.
You can't just reverse your desired trajectory back.
No. I think we have a related question in Slido, namely AI can help us deal with large quantities of data, but it doesn't know when it's right or wrong. Yeah. How do we mitigate this when using AI in the future?
So one of the things I think which is, one has to be extremely careful and rigorous in my experience. And it's quite interesting that the AI world is very experimental in the sense that one has to work out how to test these models independent of the AI metric. So independent of the internal metric about whether the model has converged and whether the model is sort of sort of, has kind of hit all of its parameter tuning and those sorts of things. You need an external test. And for us, that was switching from the UK to the Danish system. That was our very strong external test. But actually in biology, we have many other things. A really good thing is genetics, by the way, and we are bringing genetics not as additional information, but as a challenge to or a way of probing the AI network, because that's a way of testing whether our network has learned things which are really about the biology or not. And so I do think one needs to be extremely careful. But actually, I think this area, we have access to ways of getting confidence in these models, which is different from the new normal computer science setting, where often you can't bring like an orthogonal data set to the problem. So I'm quite positive, but I'd love to hear other people.
I would say, even when we evaluate a system, there is always a way to withhold 20-30% of the data and try the model on unseen data. And this is what we did in the hackathon, where we withheld and none of the participants had access to part of the data, and then we tested it on fresh data. So all the results I showed is actually completely unseen data. So that's also possible in the design of the experiment.
Okay, then I'll switch to a bit more political topic, maybe for Man as well, and the other ones at the end of the table here. Is it fair not to use data that could save patients just to keep health systems more equal and sustainable?
What?
Is that for me?
It is now.
Maybe
It's for the lawyer.
Asking the question is like answering. Well, I think that's a really unfair dichotomy to some extent. I do feel like there are always, issues that require certain levels of protection, and I think this is also related to trust. But at the same time, if we are unable to use the data that individuals have either donated or donated in terms of research, it is also a mistrust that could come out of it. And I think the idea is to be able to do this in a balanced manner, where you don't stifle innovation and you don't stop research from advancing, but at the same time keep a certain level and balance of protection.
Is there anything you would recommend to the people here who actually developed the models, what they should be doing right now or soon? Yeah.
Again, for me?
In terms of that?
Well, I mean, I was in awe listening to the research and the science behind all of this. I do feel like being part and sharing this research with colleagues that come from different disciplines, including from ethics and law, is very useful, and to continue this dialogue together.
Okay, you're going back to Slido maybe. A fundamental problem in sharing foundation models is the risk of confidential disclosures that are concealed within the embeddings. How do we overcome this while preserving privacy? I think this is again a rather technical question.
It is a question like we already have had some researchers that wanted to train some of the foundation models on the genetic data we hold. And we're just asking quite specific questions like, what is your plan to mitigate these? What are you doing? To see if the researchers that are proposing this, if they have thought about these things and if they have the capability to deal with that. And I think it's the responsibility of the data custodians to ask those questions, because otherwise people might at some point get away with murder. But it's the responsibility of us as a field to ensure that the appropriate safeguards are in place. And at the end of the day, there is that trust that actually happens, and that to a degree is inevitable, and we have to just make sure that that trust is founded.
Yeah.
I mean, I mentioned that we also, when we ran our model, we had a very good and open discussion with UK Biobank about this topic. And we actually said, we don't believe the weights are disclosing, but we can't actually be sure. We've got a 2 million parameter model. We have 500,000 people in there. You know, maybe there's a way of persuading the model to sort of regurgitate one of those individuals out. We can't do it. We can't make the model like make a precise match of one of the individual's directories that we use for training. But we can't be sure at the moment that somebody else couldn't use our model to do that. So that's why with UK Biobank, we've decided that we've already placed the weights back to, we've donated them back to UK Biobank. And at the moment, it's a protracted data aspect. You have to apply through UK Biobank to get access to it. Now, maybe in the future, we can do this. One exciting thing we are able to do is simulate a complete copy of UK Biobank from our model. So we took the UK Biobank, trained a model, simulated 500,000 people. We can train a model from that simulation. So now we're kind of 2 steps away from our data. We can then take those weights to Denmark and test it in Denmark. It's still performant in Denmark. It's slightly worse than the first model, but not that much worse than the first model. So there is opportunity here about thinking about synthetic data sets and stuff like that. But again, I think we need more research, and there may be fundamental computer science ways of asking this question. Like, is the information content in the model, you know, what is can I match the information contents in some way? It's quite hard to imagine how you precisely test that, though.
There is also, you know, a parallel discussion and research. I think if we look at the risks of like bio security and that people are like developing proteins that never have seen and using a model or vulnerabilities in the model to make harm, I think that is a little bit, if one compared to the security industry, where you have like this like 0-day bugs in the system that you are not revealing before you have a patch and I scale and fix it. I think that type of dimension also needs to be discussed in this type of situation, because there may be so that you know that there are vulnerabilities, and the big question is how do you as a community work to close some gaps, while not being transparent that there is a flaw in the model or these things before people have fixed it. So I think that is another dimension where research currently is often very transparent, that you share everything. But when we're moving into this dimension, there are things that we maybe as a community do not want to share before we have mitigated it.
Yeah, I do think we do need to, I think it's right that AI does change some of the games here, and kind of, completely new design proteins is one thing. It's worth pointing out that we do have the knowledge of very nasty bugs that are available, and ways of getting hold of very, very nasty things and doing things with it. And, the tendency has been not to regulate on the information level, but to regulate on the production and usage level. And so I do think these AI risks need to be proportionate. I'm much more worried about AI killer drones with plastic bullets, or whatever is going to happen. That seems like a near present risk that is very achievable, very doable, and very live. Whereas I think these risks are they're not zero risk, but so we do have to be proportional about where we're going to expend our discussion here. And I worry a little bit that people use this as a way of controlling that open research process for this.
Well, I think we can also learn from the ethical hacking community, where we as researchers, we can try and break each other's models in an ethical way, and then get back well, I was able to do this to your model, and we should fix it, right?
Through sandboxes,
Yeah, I mean, I think that's right, and that's why we took a cautious approach on releasing the weights. So everybody will have to do it through UK Biobank, basically. Yeah.
Okay, 1 minute. Okay, final question in the last.
Are we going to do that one?
do Skynet?
Yeah, go on, sure.
Any volunteers for that one?
Yeah, do Skynet. I want to hear the answer.
I'm not expecting it anytime soon, personally. I think some of the properties that people currently attribute to AI is just an emergent property and not an actual form of general AI or Skynet level of AI. But that's my personal view on this.
I mean, this is not our field, but I do think when you hear about what's happened in Ukraine, it's kind of remarkable, highly autonomous systems being deployed. So I personally get a little bit spooked about that technology and where it goes and who gets to use it. So I think we've got to regulate it. But that's not a biology problem.
Luckily not. Okay, I think we will wrap up at this point. good place
good
to
question,
finish.
whoever did the Skynet question.
And again, a round of applause for our speakers.
Thank you.
All right, we just have a little bit of closing remarks in just a second. We're going to let our awesome panelists de-stage. Thank you. All right, so I'm going to give a few closing remarks. We're just loading up. They're ready to go. Awesome. Okay, so that's me. So I just want to say thank you to all of the speakers. Just an amazing 2 days of talks. So thank you all, and also especially all those speakers were chosen by an amazing program committee. So another shout out to our chair, Per, and the rest of the committee who did an awesome job planning these 2 days. And of course, none of this will actually ever happen if we don't have just an amazing traveling GA4GH secretariat, many people who did a lot of work behind the scenes and are continuing to do work for the next few days as well as our local SciLife lab, particular the lead effort of Christopher Erdman. Thank you for your efforts, and the whole team walking around with those black SciLife lab shirts. So thank you to all of them for an amazing local support as well. And again, to thank, these meetings don't happen without financial support. So I really want to thank both our host of SciLife Lab, Genome Canada, Australian Genomics, Illumina, the German Human Genome Phenome Archive, and then our ongoing support from the many organizations on the right. If you weren't there last night, this was our awesome group last night. So thank you all for gathering for the picture. And then also the Destination Uppsala kindly supported our event by securing discounted hotel rates for attendees. So take a moment to complete their survey. I'll put up this code in at the end as well, so that they can assess their impact and contribute to future events like this. And then also, we, GA4GH, have our own survey that I'm sure will also be posted in Slack later, but we always want to hear feedback from all of you about what you liked, what you didn't like, what's the next great idea for next plenary or Connect meeting. So please always share your thoughts at any point in time. And then for those of you staying on for Connect, this is a little snapshot of what is coming over the next 3 days. So some of you were making comments yesterday about, gee, wouldn't it be good if all our national initiatives had a arena to organize? We have that, NIF. So NIF has been adjacent to GA4GH in its meetings, but this year we actually integrated the talks that would have been at a NIF meeting into the plenary because they really showcase all of the work that we are doing in genomics and data sharing. So that already got incorporated, and then starting tomorrow, there'll be more of a working meeting for people involved in NIF. But then we also have all of the Connect sessions that have been organized by all of you, the GA4GH community, spanning every topic possible. So I am very excited for the next few days to come, and I hope you are as well. Then, we've finally finalized the date for April Connect. It will be in Montreal. It will be the 14th through 17th of April. So put that in your diaries and plan to be there. And then I'm very excited to announce that our next year's plenary, the 14th plenary, will be in Singapore. We just had a lunch meeting about that. And in fact, I'm going to invite Shiwei and Per to come up on stage, because if you haven't been a part of the handover steps of GA4GH plenary, there's a certain process that happens every time, and that involves a certain character shown here. So I'm going to allow the official handoff of Wes from Per to Shiwei. This dude travels more than I do. Thank you, guys. Thank you for hosting an awesome meeting, and thank you for taking the plunge for the next one to come. It's going to be a blast. We want to see all of you in Singapore next year, in addition to Montreal in the spring. So we're partnering with Precise and our team in Singapore to put on an awesome meeting. And actually, this is your video, right? So I will play what's to come here, if this works. Let's see. Is it loading? Something's happening down there. We will give it another shot, maybe. Ah, there we go. It's a quiet, serene surrounding in Singapore. Imagine the sound in your head. Stargazing. Imagine floating through the sky. I can narrate here. Birds everywhere, waterfalls. Little creatures far and large. Ooh. We're going to try to avoid the F1 thing that's going to be there in October by doing it a little earlier in September. Wow, impressive. I was at the top of that. Museum nights, interesting. New things to come. Parties, that's us. You can see yourselves dancing next year. What are we eating now? Whoa. Food. Ooh. Showtime, made in Singapore. Under the lights. Are we on a roller coaster? No. Made in Singapore. All right. I promise there'll be better narration next time we show that. With that, again, plenary feedback and destination Uppsala, and then the very last thing is to welcome you to our interoperability showcase, where all our posters as well as booths from both Illumina and GA4GH will be out there. And so I encourage you. There will be beverages and light bites served. So with that, enjoy. Thank you.